Imagine you have a single, massive blueprint for an entire city. How to use that one document to build a skyscraper, then a school, and then a park? Well, our cells face this exact problem every single second. And the solution is a beautiful process called transcription. Today, we're going to dive deep into how cells read their own instruction manual, their DNA, to bring that blueprint to life.
So here's our game plan. First, we'll set the stage with the cell's big information challenge. Then, we'll zero in on transcription itself, how the copy gets made. We'll walk through the whole process from start to finish, and then explore the key differences in more complex cells like ours. And we'll wrap up with the final touches that get the RNA blueprint ready for its big mission.
All right, first up, the cell's information challenge. Think of your DNA as the master blueprint for a giant mansion. How do you use that one set of plans to build a kitchen that's totally different from the bedroom or the bathroom? They all need to look and work differently, right?
And that leads us to a really fascinating question. I mean, think about it. If pretty much every single one of your trillions of cells has the exact same DNA code, why aren't you just a giant blob of identical cells? How can a neuron in your brain and a cell on your skin be so completely different when they're working from the very same script? Well, the secret is all about selective gene expression. See, cells don't use all of their genes all the time. The key is understanding this flow of information, something biologists call the central dogma. Basically, information flows from the permanent hard drive, the DNA, to a temporary message, which is RNA. And that RNA is then used to build a functional protein. And it's that very first step from DNA to RNA that we're focusing on today. Okay, so let's get to the heart of it, transcription. I love this analogy. If your DNA is a giant, priceless master cookbook, you're not going to drag the whole thing into a messy kitchen, right? No way. You'd carefully copy a single recipe onto a small recipe card. That is exactly what transcription is.
So the formal definition. Transcription is the process of creating an RNA copy from a DNA template. It's the cell's way of making a portable, disposable instruction sheet from just one tiny section of that master blueprint.
Now, it is absolutely crucial not to get this mixed up with DNA replication. They sound similar, but their goals are totally different. Replication is like photocopying the entire cookbook, from cover to cover. It only happens right before a cell divides. Transcription, on the other hand, is like copying just one recipe, exactly when you need it. It's all about creating temporary messages on demand.
And the star of this whole show is an amazing enzyme called RNA polymerase. This is the molecular machine that actually reads the DNA and builds the RNA chain. It moves along in the same 5' to 3' direction as its cousin, DNA polymerase. But, and this is a really big deal, RNA polymerase can start a new chain all by itself. It doesn't need a primer to get it started. It just finds its spot and gets to work.
So, how does this incredible machine actually work? Let's break the process down. And we're going to start with the simpler system you find in prokaryotes, like bacteria. This gives us a fantastic foundation before we tackle the more complicated version later on.
The whole process unfolds in 3 really elegant stages. It all starts with initiation, which is all about finding the precise starting line. Then you've got elongation, which is the main event, building that RNA strand. And finally, it all wraps up with termination, where the process knows exactly when to stop and let go of the finished copy.
So let's look at initiation. RNA polymerase doesn't just land anywhere on the DNA. Nope, it looks for specific landing pads called promoters. In bacteria, these promoters have really recognizable sequences, kind of like signposts, at spots we call the -10 and -35 positions. These basically scream, hey, start transcribing right here.
Now, the bacterial RNA polymerase is actually a 2-part team. You've got the core enzyme, which does all the heavy lifting of building the RNA. But it's kind of blind. It needs a guide. That's where the sigma factor comes in. It's like a GPS unit that specifically seeks out and grabs onto those promoter sequences. Without sigma, the core enzyme would just drift along the DNA with no idea where to start.
Once initiation is locked in, we move into the second act, elongation. The sigma factor's job is done, so it pops off, and the core enzyme starts chugging along the DNA strand. It unwinds a small section of that double helix, creating what we call a transcription bubble. And inside that bubble, it reads one of the DNA strands and builds a new, complementary strand of RNA.
And finally, termination. How does it know when to stop? Well, bacteria have two clever little tricks. In what's called intrinsic termination, the RNA molecule itself has a sequence that lets it fold back into a hairpin shape. The structure literally bumps the polymerase and makes it fall off the DNA. The other method uses a protein called Rho. It latches onto the new RNA, zips along it, catches up to the polymerase, and basically just pushes it off. Pretty cool.
So that's the basic rundown in simple bacteria. Now, let's see how this whole system gets a major upgrade in complex cells like ours. This is like going from a simple go-kart to a full-blown Formula One race car.
Okay, the first big challenge for eukaryotes is just getting access to the DNA. See, our DNA isn't just floating around. It's tightly wound up around proteins in this dense structure called chromatin. So before the cell can even think about transcribing a gene, it first has to loosen up and unpack that specific region of DNA.
And the complications definitely don't stop there. Instead of one main RNA polymerase, eukaryotes have 3 specialized ones for different jobs. Plus, transcription happens safely inside the nucleus, while protein making happens out in the cytoplasm, so the two processes are physically separated. And maybe the biggest difference of all, the polymerase can't find the starting line on its own. It needs a whole entourage of helper proteins called general transcription factors.
So how does this eukaryotic team even assemble? Well, it all starts at the promoter, just like before. Many of these promoters have a key sequence called the TATA box, and the very first step is for one of these general transcription factors to find and bind to that TATA box. This is the anchor, the foundational event that kicks everything off.
Once that first factor is locked in place, it acts like a beacon. It sends out a signal that recruits all the other necessary helper proteins. They assemble one by one, building this landing platform. And only then can the star of the show, RNA polymerase II, be brought in to form the complete pre-initiation complex.
But how do we get liftoff? The whole complex is just sitting there. Well, the final factor to join the team has a very special job. Think of it like a starter motor. It adds a bunch of phosphate groups to the tail of the RNA polymerase. This chemical kick gives the polymerase the energy it needs to break free from the complex and finally launch down the DNA to start elongation.
But believe it or not, the story still isn't over when transcription stops. In eukaryotes, that freshly made RNA is like a rough draft. It needs some serious editing and processing before it's ready to be used. This is how we get from the draft to the final, polished blueprint.
That initial transcript is called a pre-mRNA, and it is definitely not ready to leave the nucleus yet. It has to go through 3 major modifications to become a mature functional messenger RNA, or what we call an mRNA.
Here are those 3 key steps. First, a protective 5' cap gets added to the front end of the RNA. This acts like a helmet, protecting it from being destroyed and helping the cell recognize it as a real message. Second, a process called splicing happens. This is where all the non-coding junk sections, called introns, get snipped out, and the important coding parts, the exons, are stitched together. And finally, a long string of adenine bases, the poly-A tail, is added to the back end for stability.
And with that, we have our mature mRNA blueprint, ready for action. We've seen how the cell meticulously and carefully copies a single recipe from its vast genetic cookbook. But this incredible journey of information is only half the story. The next giant question is, how does the cell's construction crew actually read this blueprint to build the proteins that make you, well, you? Now that is a story for another time.